your notes on the cell cycle and mitosis. All the images that I'm using are labeled for reuse. They're from commons.wikimedia.org. Before we get into the cell cycle and mitosis, let's review a little bit about what we know of chromosome structure and DNA. So remember, we talked about this before. We've done our DNA unit and you figured out the structure of DNA. It's this double helix with 2 strands. And those base pairs are A, T, G, and C. That DNA gets wrapped up a little bit and it's in the form of chromatin. So here it's wrapped up loosely. Remember that it's in the form of chromatin when the cell wants to make proteins, when it's doing its normal stuff, its normal cell processes. But then when the cell wants to pack up and move, it has to bundle its DNA up even tighter in the form of chromosomes. So I compared this to you living in your house. Right now, all of your stuff is spread out so that you can find it, you can get to it. But if you're going to move, you would pack up all your stuff into boxes. That's what DNA does when it makes a chromosome. It packs all of its DNA up so that it's ready to move its DNA around. All right, so you need to be writing this down as we go here. So you should be writing chromosomes are composed of DNA and protein. The proteins are called histones, so you don't have to know that. And the DNA is wrapped up tightly around these histones. Most of the time when you see a chromosome, you'll see it attached in the middle. It will kind of look like an X. These are 2 chromosomes that are attached to each other. That's an identical copy. Remember, we just learned about DNA replication. So here, this is a replicated copy of the DNA. This would be an old strand of DNA and a new strand of DNA from DNA replication, and a new strand of DNA and an old strand of DNA. And they stay attached to each other here in the middle during cell division. Those together are called sister chromatids. You can think of them as twin sister chromatids because they should be identical copies. They're duplicated chromosomes connected at the centromere. You may have to pause this video so you can write all these notes down. The first phase of the cell cycle is interphase. The cell spends about 90% of its time in this phase. That number is going to vary depending on what kind of a cell it is, but about 90%. Interphase is 3 main processes. The first one is a growth phase. The cell grows, it carries out its normal functions, it makes the proteins it's supposed to make, it does whatever it's supposed to do in your body. It's going to synthesize proteins, it's going to repair. Then you have this S phase. The S phase is when you do DNA replication. Remember, that's what we talked about in the last set of notes. So you synthesize, S for synthesized DNA. Then you have a little bit more growth in the G2. It's the second growth phase. So here now the cell knows it's going to have to go into mitosis, and so it has to double everything. It'll have to make like more mitochondria and more endoplasmic reticulum and get bigger and prepare to divide. Then the mitosis part of the cell cycle, you notice that is in here too, this little blue part. After G2, the cell could go into mitosis. Mitosis is a division of the nucleus. You use mitosis when you want to make identical body cells. Some really simple organisms use mitosis to reproduce. Like bacteria use a type of mitosis to make another copy of themselves. You don't do that. You do meiosis, which we'll talk about later. Because you have 46 chromosomes, you will make 2 daughter cells at the end of mitosis that also have 46 chromosomes. So mitosis, the chromosome number doesn't change. You should make an exact copy of the cell with exactly the same number of chromosomes at the end. Here I've got this karyotype. This is a picture of DNA. You can see this is 23 pairs of DNA, which would be 46 chromosomes. We'll get into genetics next. You can see that the XY, some of you know that that would be a male, and then the XX is a female. And you've got 22 pairs of other chromosomes. After mitosis, now we're going to go back and talk about all the phases of mitosis, but the next main step after mitosis is cytokinesis. Cyto means cell, and kinesis means to move. So it literally means to move the cell. It's dividing the cell in half. This is the last stage of the cell cycle and splits the cytoplasm in half. This usually happens at the same time as the last phase of mitosis, which is telophase. So you can see the cell, this is an animal cell. In an animal cell, the cell membrane is going to pinch in and slowly separate into two separate cells. All right, so starting mitosis. In mitosis, one of the first things that happen is those chromosomes start to become visible during cell division. We will hopefully look at these cells under the microscope. So you can see this one here where everything is all fuzzy. That's the DNA inside the nucleus in the form of chromatin. It's all spread out. You can't really see any definition in the DNA. Remember a little basic here. This is the cell membrane going around the cell. This is the cytoplasm inside. This is the nucleus. But here in some of these other cells, this cell and this cell and this cell, you can start to see thicker bands of DNA. Those are the chromosomes. The first cell of mitosis is pretty long, so you can see like there are different phases within that first phase. Another key idea here is that you have homologous chromosomes. If we go back to that karyotype, you can say that you've got 22 pairs of chromosomes. Well, those pairs are homologous chromosomes. You get a copy of chromosome number 1 from your mom, and you get a copy of chromosome number 1 from your dad. And then the same thing with all of your chromosomes. Those are homologous. Homologous means same chromosome. They're not identical like sister chromatids, but they have similar information on them. So here, this is number 1 chromosome, and this is number 1 chromosome from dad. They should have, like if this has eye color on it, this chromosome will have eye color on it. If this chromosome has the gene for making hemoglobin, this chromosome is going to have the gene for making hemoglobin too. They might not be exactly the same gene because your parents might have different versions of those genes. So here my picture, chromosome 1 and 2 are homologous, and 3 and 4 are homologous. And I used the color to represent whether they came from mom or dad. So I made the red ones from mom, 1 and 3 would come from mom, and then 2 and 4 came from dad. Here's my little picture of the cell cycle. So we've got the first part of the cell cycle, remember, is the growth phase, G1. We've got growth, you've got protein synthesis. Their cells are making the proteins they're supposed to make, and they're carrying out their normal functions. Sometimes cells just don't go any farther than this. They go into something called G0, where they are just suspended in that state, where they're just a mature cell and they will never divide again, like nerve cells. We're never going to synthesize their DNA and go into mitosis. They're just going to stay, at least some of them, stay in this G0 phase. Your other cells, though, are going to go on, and at some point, eventually, they're going to do DNA replication. But there are little checkpoints in here. There's a checkpoint where your cell stops and it checks out the DNA and it makes sure that there's not any damage. If there's damage, the cell might kill itself. It's called apoptosis. So the cell might, and that's actually a good thing, because if you skip these checkpoints, that's when you end up with things like cancer. So if you go ahead and replicate your DNA, even though there are problems with it, you might end up with a cancerous cell. Or cancerous cells will end up being able to skip these checkpoints, and that's why they can keep on replicating, even though they're a cancer cell. So then if your DNA is okay, you will go into the synthesis phase, replicate your DNA. You'll go into G2, where your cells duplicate all their organelles and get ready to divide. And then here there's another checkpoint, an apoptosis checkpoint. Is the DNA okay? Are the chromosomes ready for mitosis? If they are not, now the other option is that your cell tries to fix it. But if it can't fix it, then it's apoptosis. So the cell destroys itself. Sounds bad. It's actually good. You're doing that with cancer cells in your body. Then you'll start mitosis, and there's another checkpoint in here. We haven't talked about spindle fibers yet, but there's a spindle checkpoint where you make sure that all your sister chromatids are correctly attached to the spindle fiber, which we'll talk about in a minute here. We'll get into some of those checkpoints again later when we talk about cancer. All right, so mitosis, the basics, you've got a division of the nucleus. You make two hopefully identical nuclei. It makes a diploid cell. Di means two. Ploids means sets of chromosomes. So diploid or diploid means you have two sets of chromosomes. In a human cell, the diploid number is 46. So we should end up with a cell with 46 chromosomes at the end. Some people remember this by remembering mitosis, like you make your toe cells with mitosis. You make all of your body cells with mitosis. Fancy word for body cells is somatic cells. All right, phase one, there are 4 phases of mitosis because cytokinesis isn't technically a phase of mitosis. There are 4 other phases. There, you can remember PMAT for short. Prophase is first. Pro means before, or first phase. A lot of things happen in prophase. You'll see if you read some textbooks, they break prophase down into multiple phases. The 4 basic things you need to remember is that this nuclear membrane starts to go away. So here it's shown as a dotted line because that nuclear membrane starts to go away. Remember, cell membrane on the outside. It's not going to go away, but this membrane does. Those chromosomes start to coil up, so they're starting to look like X's. And you do get a spindle fiber. Here, the spindle fiber is here. And eventually, by the end of prophase, it'll attach to those chromosomes. So here, this is going to be an earlier prophase where the spindle fiber hasn't quite formed yet and it's not attached to the chromosomes. When we look at these under the microscope, we're going to look for those chromosomes starting to show up. So it's not this blurry bundle of DNA in the nucleus, but they're defined bands of chromosomes. The second phase of mitosis is called metaphase. PMAT. I also remember metaphase middle. The chromosomes in metaphase are lined up in the middle of the cell along the equator, or what's sometimes called the metaphase plate. You can see it's a single line of chromosomes. This is going to be important later when we go through meiosis. It's not a single line of chromosomes. Of course, this is a cartoon picture here, but when you look under the microscope, it's not really pretty like that, but you do have this thick band of colored DNA going down the middle of the cell. All right, anaphase. I remember anaphase away. Those chromosomes are starting to move away from each other or apart. So those sister chromatids have split. You've got one sister chromatid going this way, one sister chromatid going the other way. And actually, once those chromatids split, they're called chromosomes again. This is the cell we were looking at in the last slide, the metaphase cell. All these other cells, remember, are an interphase. But here in this cell, you can see you've got this line of chromosomes and you got that line of chromosomes. And they look like they're being pulled away from each other because they get pulled by the middle. So they kind of flatten out and the ends drag back as they get pulled by the middle. So that's why these ends are hanging back toward the middle. And then the last phase of mitosis is telophase or telophase, depending on who you ask. You could remember TE for the end. And this phase is the opposite of prophase. So prophase, the nuclear membrane broke apart. Here, that nuclear